All right, so now we're on our last section of talking about homeostasis. We're going to talk a little bit about respiration. And so I want to make sure that we understand we kind of use this term in a couple different ways, at least in zoology. Earlier, we talked about cellular respiration. So we talked about glycolysis and the Krebs cycle and the electron transport. And we sometimes call this metabolism. Molecules being harvested and broken down to harvest their energy. In this lecture, we're going to be talking about external respiration, exchanging gases with the environment. So, use the context to make sure you understand the difference between these two. And so, to start with, there's lots of different respiratory organs that you'll find throughout the animal kingdom. Some animals use direct diffusion. They don't really have any organs. They just allow the gases to diffuse in and diffuse out. There are tracheal systems, gills, and lungs. And we'll talk about each of those in a little bit more detail. In lab, we talk about flatworms. They don't really have respiratory organs, but they're flat. They've got a very high surface area, very high surface area to volume ratio. And so they can just allow gases to diffuse in and then out of their cells. The protozoans, the sponges, the cnidarians, the segmented worms, all these organisms don't really have respiratory organs. They just allow gases to diffuse in and diffuse out. But again, these, along with the flatworms, they have higher surface area to volume ratios. There are vertebrates who do some respiration just across their skin. They allow gases to diffuse in and diffuse out. Amphibians and reptiles in situations, or sometimes they use this as a supplemental respiration, but sometimes when they're small, this is the only way they respire. But again, what's common to all of these? They all live in wet environments. You need that wet environment in order to have gas exchange, and so that's where you can see a lot of this diffusion. But it's also pretty inefficient, and so having actual organs that are dedicated to respiration is more efficient and allows you to be more active. And so you see things like tracheal systems that have evolved in arthropods. And these are just branching tubes called tracheae. And so these are just like tubes that are stuck all throughout the body that just have an opening to the outside. So it's still, gases just sort of moving in and moving out, but instead of like having to move across the body surface, they have these tubes to direct the gases into the body and out of the body, and that makes it more efficient. And it's very efficient and very effective, and it's one of the reasons that like insects are so successful. And so the openings are often called spiracles, where the tracheae open to the environment, and those openings can be controlled. And so, if you've got this opening, you can lose water, and sometimes if you have to conserve water, you can just close these off so you don't have as much water vapor leaving. And so these just sort of are like tubes that go directly to the other organs and directly to the inside of the body, and they're all over the body. So you don't really need to carry oxygen and gases in the blood or in the hemolymph, because it's not like like in us, where we take in the gases in one spot and then we have to move them to everywhere else in the body. So that's why we need blood. Here, you've got these tracheae all over the body, and so the gases can just diffuse straight into where they're needed. So you don't really need the circulatory system to carry gases as much. So that's why you can get away with an open circulatory system to some extent. Okay, gills are often a specialized organ for gas exchange, and when we look at mollusks, we see a lot of them have gills. A lot of arthropods, especially in their larval stage, will have gills. So a lot of insects that the larvae hatch in the water and their first life stages in the water, they'll have gills for gas exchange. Of course, fish also have gills for gas exchange. And if you look at fish, you can see how, again, you have the combination of the respiratory organ and other anatomical organs and behaviors that all combine to increase efficiency. And so here we're looking at the mouth of the fish and the opening of the fish called the buccal cavity, and then you've got the gills. And so what they're showing you here is that when the mouth is open, the operculum is closed. So the operculum is the cover on the back of the buccal cavity where water exits, and it covers the gills. And so those are closed when the mouth is open. And so by increasing the volume of this buccal cavity, you create an area of low pressure. And so you've got high pressure outside the fish, low pressure inside the buccal cavity, so water is going to flow in. Then the mouth closes, the buccal cavity is squeezed shut, you decrease the volume, and so you increase the pressure in the buccal cavity. Now there's higher pressure inside the mouth relative to outside, but those opercula open up, and so that forces water. Now you've got high pressure inside the mouth relative to outside, so water's going to flow out, but it's going to flow out the other openings, and it's going to flow across those gills. And so that's what they're showing you here. So when one is open, the other's closed. And you'll remember at those gills we talked about, that's where you have a countercurrent mechanism. So here we're seeing how the water is forced in one direction past the gills, the blood is pumped in the opposite direction, and there you can exchange, get rid of carbon dioxide and pick up oxygen. Now I'd like to take a side note. I think this is kind of not a very good image, and maybe I'm missing something here, but to me this looks all wrong. It looks like we're looking down on the fish, like a dorsal view of the fish, because of the way the gills are sort of organized. But look at how the mouth opens. The mouth does not open like this in a fish. The mouth opens like this in a fish. I don't know, maybe I'm missing something here. But if maybe somebody who understands this better can explain it to me. But to me, it looks like the mouth and the gills are sort of at 90 degrees to each other relative to a fish. But it doesn't matter. It's just me being picky. You get the idea. You get the idea of the mouth opening and the opercular covers closing, and vice versa, and how the pressure makes water move. Okay. So then, last thing we talk about, of course, are lungs, which you're going to find in mammals, of course, but reptiles, amphibians have lungs. But you also have some fish. So lungfish have a swim bladder that has evolved into a lung, and some of them are obligate air breathers. That it's a fish that must make it to the surface to suck air. And so, you know, this is a fish that you could theoretically drown just by holding its head underwater. If you look at lungs, you can see that a lot of the lungs have an increased complexity. They're trying to increase their surface area to maximize the exchange of gases in the environment. So you got a lot of surface area in there. But they can be less efficient than gills because you've only got one opening. You've got the trachea that allows gas to come in and out the same, you know, in the same opening. Whereas in the gills, it comes in one way, comes out the other way, which is more efficient. And so, you know, you inhale and exhale through the same opening. And so you inhale and you're bringing in fresh air, but then when you exhale, you cannot bring in fresh air. And so it's actually, you know, for all its advantages, it's a little bit less efficient. But then you see adaptations that, for example, in birds that take this into account, and you've got these secondary air sacs that aren't vascularized. They don't have blood vessels. There's not any gas exchange. They just store fresh air. And so when you breathe out, now this fresh air can go past the alveoli and the places where oxygen is exchanged. And so you're exchanging gases both on the intake and on the inhale and on the exhale. So that's pretty cool. Okay. So we want to combine now this respiratory uptake with the circulatory system, because the two kind of go hand in hand. You know, we got to take in the gases, then we use the circulatory system to move those gases around. And this is the last thing I want to talk about. And so moving oxygen around the body is obviously a pretty big deal, but oxygen doesn't dissolve well in water. And so a lot of organisms have hemoglobin, or a very similar molecule that's designed to carry oxygen in the blood. Since it doesn't dissolve well, we're going to bind it to a molecule and use that molecule to carry oxygen around. And in us, it's hemoglobin, which we've talked about several times now. It's a big protein. It's got quaternary structure, and it carries both oxygen and carbon dioxide. So it's very good at carrying oxygen to the tissues and carbon dioxide away from the tissues. So when oxygen diffuses into the blood, it doesn't dissolve in the fluid of the blood. It binds to the hemoglobin. The hemoglobin. Hemoglobin. What's wrong with me? It binds to the hemoglobin. Well, consequently, the partial pressure of the blood, the partial pressure of oxygen in the blood doesn't change, because the amount of dissolved gas in the blood doesn't change, because the oxygen is not really acting like a dissolved gas. It's coming in and it's binding to a molecule. So it's acting like something that's dissolved in the blood. It's acting like a particulate. You know, it's acting like a hemoglobin molecule, not like a gas. So the gas partial pressure doesn't change, which means that the difference in partial pressure stays the same, which means that oxygen can keep diffusing into the blood. So let me try and explain that a little bit better than what I just did. Here we've got, you know, a capillary wall. So this is inside, is it a fish gill, or this is, you know, inside the alveoli of your lungs. Again, we've got water and blood because you need an aqueous environment for these exchanges to take place. And so, say the water or the air in your lungs is 100% saturated with oxygen, very high partial pressure of oxygen, but the blood has very low partial pressure. Well, again, that's why it's at the lungs or the gills, because you're trying to pick up oxygen. So you've got a very big difference in concentration, so you're going to get diffusion of oxygen into the blood, which is what you want. But when that oxygen diffuses into the blood, it binds to hemoglobin. So it's as if no oxygen diffused at all. You see that the partial pressure is unaffected. So you still have this very large difference in partial pressure, which means you're still going to have a lot of oxygen diffusing in. So this is another way. Earlier we talked about a countercurrent mechanism for maximizing oxygen uptake. This is also something that helps to maximize oxygen uptake. By binding to hemoglobin, you keep the partial pressure in the blood low, which allows for more rapid diffusion of oxygen into the blood. So that's another advantage of having hemoglobin. Okay, and so again, when we look at hemoglobin, we talk about its complicated shape. You've got 4 subunits that all come together. Like any other protein, or most proteins anyway, shape influences function, and it's really important in hemoglobin. Each of these subunits can hold an oxygen, so every hemoglobin can hold 4 oxygen oxygen atoms. And when we talk about hemoglobin, we say that it has an affinity for oxygen. And we use affinity like you always use, affinity, like how much do you like something? Do you have an affinity for it? Well, hemoglobin's affinity for oxygen kind of runs hot and cold, right? Sometimes it loves oxygen and it wants to grab it and hold on to it and love it forever. And sometimes it's like, and it'll just let it go, right? And so the affinity of hemoglobin for oxygen can change, but it's the shape of hemoglobin that determines this affinity. Change the shape, change the affinity. Which is what I just said right there. And so how does this work? If there's no oxygen available, the hemoglobin is shaped in such a way that all the binding sites are hidden. And so it doesn't really have an affinity for oxygen because it's got nowhere to bind oxygen. But if oxygen is present, eventually one oxygen will find a binding spot and will bind to the hemoglobin. And as you get more oxygen, it's just more likely that that oxygen is going to find somewhere to bind. It's going to find an open binding spot. Again, you know, proteins are constantly, they're not static. Their temperature changes. They're constantly changing shape. But in general, if there's no oxygen, those binding sites are covered up. But if oxygen is present, eventually an oxygen is going to find a binding site and is going to bind to hemoglobin 1. But when this happens, that causes hemoglobin to change shape. And when it changes shape, it opens up the other 3 binding sites. And so then very quickly, 3 more oxygens can come in. And so that first oxygen binding unlocks the door and allows 3 more to come in. And so at this point, hemoglobin has as much oxygen as it can carry, and we say the hemoglobin is saturated. And then the opposite is kind of true as well. Once hemoglobin loses, a saturated hemoglobin loses 1 oxygen, that changes the shape enough that it changes the affinity and it quickly drops the other 3. And so all this means that hemoglobin is kind of an all or nothing kind of thing, right? And so either, you know, it tends, if it binds an oxygen, then it's going to bind 3 more oxygens and it's going to be saturated. And if a saturated one loses an oxygen, then it's just done with oxygen and it drops all of them, right? You get kind of an all or nothing thing. So looking at how many hemoglobins are saturated kind of gives you a insight into what's going on in the blood. And so when we want to look at this, we can kind of make a graph and we can kind of look at how the amount of oxygen changes how many of the hemoglobins are saturated. And so this is kind of a classic graph or a classic way to look at this, and you've got this in your book too. But study this graph and make sure you understand what it is that we're graphing here. If you look on the y-axis, we've forgot the percent of hemoglobins that are saturated. So it's how many of the hemoglobins in your blood are carrying their full load of oxygen. Remember that, you know, it's kind of an all or nothing thing, right? Either you're not carrying any oxygen or you're carrying you're carrying as much oxygen as you can. And so by looking at the percent of hemoglobins that are saturated, that gives you an idea of what the oxygen, how much oxygen is in the blood, what's going on in the blood. So that's what we're graphing on the y-axis. On the x-axis, we're graphing the partial pressure of oxygen in the blood. Basically, how much oxygen is in the blood. So we're looking at, as oxygen increases in the blood, how does that affect how much oxygen is being carried by hemoglobins? And so when you do that, you get a classic S-curve. And so when oxygen is low, down near the origin, the percent of hemoglobins that are saturated is also low. There's no oxygen. How can hemoglobins be saturated, right? As oxygen increases in the blood, then you start to see more and more of your hemoglobins are saturated. So as you start to see oxygen molecules in the blood, you know, some hemoglobins are going to grab one. When they grab one, then they open up and they grab 4 more, and now you've got a saturated hemoglobin. And so as the oxygen increases, you can see the percent of saturated hemoglobins increases, and then increases very rapidly. But then you see that it asymptotes. You see that as oxygen gets higher and higher, eventually this curve levels off. Well, why does that curve level off? Well, at some point, all the hemoglobins are saturated, right? And increasing oxygen, you can't have, and you know, all the hemoglobins are carrying all the oxygen they can carry, so adding more oxygen doesn't affect anything because they're already got their hands full, right? So that's why this graph is shaped like it is. But this is all influenced by that idea that you know, once a hemoglobin binds to 1 oxygen, then it opens up and can grab 3 more very quickly. And this also works in reverse. And so if you've got a lot of oxygen in the blood, then most of your hemoglobins are saturated with oxygen. But as the oxygen drops in the blood, then, you know, again, it's more likely for hemoglobin to drop in oxygen, and once it drops one oxygen, it drops all of them. And so as oxygen drops, then the hemoglobin saturation drops very rapidly. And that's what I'm saying here. It works in both directions. But once oxygen starts to drop in the blood, the hemoglobins rapidly dump their oxygen. Okay, so now let's tie this again to the respiratory system. And let's talk about blood chemistry at both the lungs and at the tissues. So the lungs, of course, is the respiratory organ. The tissues is, the blood's carrying gases to the tissues. At the lungs, in general, what do we have? These arrows indicate we've got high oxygen and low carbon dioxide, right? That's because you're breathing in oxygen, breathing out carbon dioxide. At the tissues, you have just the opposite. You have low oxygen and high carbon dioxide because the tissues are consuming oxygen and giving off carbon dioxide, right? So you kind of got opposites at the tissues versus the lungs. Well, where is this coming from? All of a sudden, we've been talking about oxygen, oxygen, oxygen. Now I'm talking about carbon dioxide. Well, you can't talk about one without talking about the other. But in this example, it's important to understand what carbon dioxide is doing in the blood. Carbon dioxide is influencing the pH of the blood. Where am I going? Well, stick with me. This is why we talked about things like pH earlier, right? Look at this equation. I want you to know this equation because it shows up everywhere in biology. Carbon dioxide reacts with water to form carbonic acid, H2CO3. Then carbonic acid, it's an acid. So what do acids do? They break down and give off protons. So that carbonic acid breaks off and gives off hydrogen ions, protons, plus bicarbonate, HCO3. Now this HCO3 can also break down and give off more hydrogen ions, but this is good enough for now. So when CO2 dissolves in water, you make carbonic acid, which is an acid. But what do acids do to the pH? They lower the pH. And so that's something that's very important to what we're talking about. The presence of carbon dioxide means that the blood pH is lower because of this reaction. Well, why do we care in this case? Why do we care about pH? Remember, what are some of the things that can influence the shape of a protein? We said temperature is a biggie. We said salts can influence the shape. And we said pH can influence the shape of the protein. This is an example of that. pH changes the shape of protein, the shape of hemoglobin. Well, what do we say about the shape of hemoglobin? The shape of hemoglobin influences its affinity for oxygen. So by changing, the CO2 levels can influence the pH, which can influence the shape of hemoglobin, which can influence its ability to carry oxygen. That's how these things are all related. And so again, I just said that if you change the shape of hemoglobin, you change its affinity for oxygen. And so this curve that I showed you earlier, let's say that that's at a pH of 8, just a little bit higher than neutral. That's the curve we're looking at. But if we've got blood that's at a pH of 7, it shifts the curve to the right. And this is called the Bohr effect. And so now compare these two curves and think about what they mean. At the green curve, at the pH of 7, does the hemoglobin have a higher or lower affinity for oxygen? Think about it. Well, at the pH of 7, the hemoglobin has a lower affinity for oxygen. It doesn't bind as well to oxygen. How do I know that? Well, pick a particular oxygen level on the x-axis and look at the two curves. And so if I pick a spot, somewhere, a little ways up on the x-axis where I don't have a whole lot of oxygen in the blood, you can see that if the oxygen is low in the blood at a pH of 8, well, some of the hemoglobins are still grabbing some of that oxygen. But if the pH is 7, none of the hemoglobins are grabbing oxygen. And so that means that the hemoglobins have a lower affinity for oxygen. And as the oxygen climbs, you see at a pH of 8, now the hemoglobins are grabbing oxygen like crazy, and they're getting saturated like crazy. But at a pH of 7, the same hemoglobins are like, okay, they don't have quite the affinity. And so by lowering the pH, you lower hemoglobin's affinity for oxygen, which means, again, the opposite. If you've got lots of hemoglobins that are saturated and they're holding on to their oxygen, and then you drop the pH, now their shape changes a little bit. Now they don't have so much affinity. And so they're going to let go of all that oxygen. And so hemoglobin cannot hold oxygen as well when the pH is low. That's the Bohr effect. And so if the pH drops, hemoglobin will drop oxygen. Well, where does the pH drop? The pH drops where carbon dioxide is high. Well, where is carbon dioxide high? Carbon dioxide is high at the body tissues, which need oxygen. Well, that's awesome. So the net effect is for hemoglobins to drop oxygen right where it's needed. So you see how all these things work together to control where the oxygen is dropped off. And so here's a figure from your book kind of showing you the same thing, showing you that at lower pHs, hemoglobin does not hold oxygen as well. Those lower pHs occur where CO2 is high. Well, where do you need oxygen? You need oxygen where CO2 is high. So that's the Bohr effect, and it's pretty cool. So the S shape of the curve is sort of due to oxygen's effect on the shape of hemoglobin. You know, that all or nothing thing, right? And so once oxygen increases, the percent saturated hemoglobins increases very quickly. The curve shifting to the right, that's pH's effect. That's the Bohr effect on hemoglobin shape and its ability and its affinity for oxygen. And so, you know, this is, again, this is one way that you can control where that oxygen is being dropped off. And so this is how, like, fish can inflate their swim bladder and all kinds of cool stuff. All right. Okay, so that was just a brief introduction into respiration and tying it back to the circulatory system. That kind of wraps up our section on homeostasis. We've talked about several different things. So, as always, let me know if you've got any questions. I'll see you later.